He realized that he was through with commercial programming when he received Microsoft's bulky 12 CD-ROM package for developers of programs for Windows 95. I was thinking to myself that the whole human genome could fit on one CD-ROM and didn't change every 3 months. Confident in May that he had a good way to crack the much-talked-about assembly problem, he induced his university to let him borrow 100 PCs recently bought for teaching purposes. He then embarked on a 4-week programming marathon, icing his wrists at night to prevent them from seizing up as he turned out computer code by day. His deadline was June 26th, when the completion of the rough draft was to be announced. The program finished, he set his 100 PCs to work, and on June 22nd, his gang of PCs solved the public project's assembly problem. Myers at Celera cut it even closer, completing his assembly on the night of June 25th. Then came June 26th, 2000. Bill Clinton at the White House and Tony Blair at 10 Downing Street simultaneously proclaimed the first draft of the HGP complete. The race was called as a tie, the honors to be shared equally. Happily, the opposing parties managed to put the bad feelings behind them, for the morning at least. Clinton declared, "Today we are learning the language in which God created life. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal." Grand words for a grand occasion. It was impossible not to feel some pride in an accomplishment that the press promptly compared to the first Apollo moon landing, even if the official date of the triumph was somewhat arbitrary. The sequencing was by no means over, and it would be more than 6 months before the scientific papers summarizing the genome were published. It has been suggested that the timing was dictated not by the HGP's timetable, but by Clinton's and Blair's schedules. Overlooked in the blaze of White House publicity was the fact that the object of celebration was but a rough draft of the human genome. Much work remained to be done. In fact, the sequences of only 2 of the smallest chromosomes, 21 and 22, were reasonably complete and had been published. And even these could not boast unbroken chromosome tip-to-chromosome tip sequences. As to the other chromosomes, some of their sequences were riddled with gaps. Since that big announcement, sights have been set on a new deadline of April 2003 for filling in the gaps and securing a full, accurate sequence. Some small regions, however, have proved literally unsequenceable. And in practice, the goal has become to obtain an essentially complete sequence. At least 95% of the sequence finished with an error rate of less than 1 in 10,000 bases. One of those responsible for coaxing the international herd of sequencing centers over the final hurdles was Rick Wilson, the bluff Midwesterner who succeeded Bob Waterston as head of the Washington University Center. Quality control is the name of the game, so each chromosome has been assigned a coordinator to oversee progress and ensure that his or her charge meets the project's overall specifications. Occasional glitches occur. For example, an errant piece of rice sequence crept mysteriously into one submission to the database, but screening procedures have proved effective in removing such contaminants. As I write this, the Human Genome Project is well on course to being essentially complete by the April 2003 deadline, which is also the 50th anniversary of the publication of the double helix. The Human Genome Project is an extraordinary technological achievement. Had anyone suggested in 1953 that the entire human genome would be sequenced within 50 years, Crick and I would have laughed and bought them another drink. And such skepticism would have still seemed valid more than 20 years later, when the first methods for sequencing DNA were finally devised. Those methods were, to be sure, a technical breakthrough, but sequencing was a painfully slow business all the same. In those days, it was a major undertaking to generate the sequence of even one small gene a few hundred base pairs in length. And now, here we were, just another 25 years further on, celebrating the completion of some 3.1 billion base pairs of sequence. But we must also bear in mind that the genome is much more than a monument to our technological wizardry, astonishing though that may be. Whatever its immediate political motivation, that White House celebration was perfectly justified in hailing the possibilities of a marvelous new weapon in our fight against disease, and even more, a whole new era in our understanding of how organisms are put together and how they operate, and of what it is that sets us apart biologically from other species. What, in other words, makes us human. Chapter 8. Reading Genomes. Evolution in Action. I used to wish that when the human genome was finally completely sequenced, it would turn out to contain 72,415 genes. My enthusiasm for this obscure number stemmed from the Human Genome Project's first big surprise. In December 1999, sandwiched between two major sequencing landmarks, billions 1 and 2, came the first completed chromosome, number 22. Although a small one, constituting only 1.1% of the total genome, chromosome 22 was still 33.4 million base pairs long. Now, this is our first glimpse of what the genome as a whole might look like. As one commentator for Nature wrote, it was like seeing the surface or the landscape of a new planet for the first time. Most interesting was the density of genes along the chromosome. We had no reason to believe that chromosome 22 would not be representative of the entire genome, so we expected to find about 1.1% of all human genes in its sequence. That is to say, given the standard textbook estimate of about 100,000 human genes in total, we should have expected to see about 1,100 of them on chromosome 22. Almost exactly half that number were found, 545. Here was the first big hint that the human genome was not as gene-rich as we had supposed. Suddenly, the human gene count was a hot topic. At the Cold Spring Harbor Laboratory Conference on the Genome in May 2000, Ewan Birney, who was spearheading the Sanger Center's computer analysis of the sequence, organized a contest he called Gene Sweep. It was a lottery based on estimating the correct gene count, which would finally be determined with the completion of the sequence in 2003. The winner would be the one who had come closest to the right answer. That Birney should have become the HGP's unofficial bookie wasn't entirely surprising. Numbers are his thing. After Eton, he took a year to tackle quantitative problems in biology, while living in my house on Long Island. A far cry from trekking in the Himalayas or tending bar in Rio, just two more of the likely ways a young Briton might spend the gap year before university. Birney's CSHL work yielded two important research papers before he even set foot in Oxford. Originally, Birney charged $1 per entry, but the price of admission to the pool increased with every published estimate that brought us closer to a final count. I was able to get in on the ground floor, putting $1 down on 72,415. My bet was a calculated attempt to reconcile the textbook figure 100,000 and the new best guess of around 50,000, based on the chromosome 22 result. The exact count is unknown as I write, but with every passing month, my bet appears more and more wildly over the mark. Looks like I'll be $1 down on the genome. Perhaps the only question to generate as much idle speculation as the gene count was that of whose genes we were sequencing. The information was in principle confidential, so money was not going to change hands on this one, but many wondered all the same. In the case of the public project, the DNA sample we sequenced had come from a number of randomly selected individuals from around Buffalo, New York, the same area where the processing work, isolating the DNA and inserting it into bacterial artificial chromosomes for mapping and sequencing, was taking place.